So first of all, thank you very much for those of you who have joined today, and also for those who listen to the recording online. So today we were keen to talk about what we describe as advanced cell culture, but essentially what it is growing cells in the laboratory from patients for a prolonged period of time, and specifically with a focus on lung medicine. So I suppose to start off with, you'll be aware that lung medicine is a key theme of the Biomedical Research Centre in Southampton. And why is it important? I mean, so many of our hospital admissions and so much of the morbidity in Southampton and the region relate to respiratory medicine. So I was on the wards over Christmas, and we had over 120 respiratory inpatients, and that's pretty standard for us. And so out of a hospital of 1,000 beds, over 1 in 10 beds are being taken up by people who have got respiratory disease. And so it's a big problem, it's a big challenge, and if we're going to help people live longer and healthier lives, we need to understand the underlying drivers of respiratory medicine and respiratory disease much better. And in traditional biomedical research, one would use mouse models or small animal models. And so you would take a mouse, you might genetically modify it, and you might study the disease process in the mouse. But emerging theme really over the last 20 years or so is that not only do mice not reflect what's going on in human patients that well, and so you might develop a drug that works really well in a mouse and then it fails in patients. Also, you can do things probably much better and more efficiently and without using animal models, using human cells. And so one takes cells from patients and you grow them in the laboratory. And hopefully, not only is that more cost effective, then when you actually move on to the clinical trials, you have a higher success rate because the drug has been developed on the human cells. But the reason for today's session really is centered on the fact that you can do more and more with human cells. You can grow them for a long time. You can genetically modify them so that they become immortal, and so you grow forever. So I think we do start coming on to some genuinely important and sort of, maybe troubling is the wrong word, but an area where we need to get feedback from the community of people we're trying to help, because I think some people may be happy with saying, well, you've taken some cells at a procedure, they're yours to do what you want with, if that may help me or people in the future from this condition. But I think other people equally might go, well, they're my cells and I want to know exactly what you're going to do with them in the laboratory. And so that's my sort of preamble. I'm going to hand over to Mirella now, who's got a few slides just to give a little bit more detail. And then I think we'll open it up for discussion. But also, because this is a recorded session, I think it'd be really great if we could have some email comments back from people who follow this, because as we develop the work programme, which is funded for the next 5 years, we want to be absolutely sure that what we're doing is agreeable and acceptable to the community whose cells we may be asking to use during these experiments. But Mirella, over to you.
Thank you, Paul. Okay, and now I will share my slides.
And I should probably say actually that Mirella is the senior scientist who's leading on these experiments, and so I'm a clinician and I walk the wards and I do some research, but it's actually Mirella who's in charge of what's going on in the laboratory.
Okay, now can you see my slides in presentation mode? Perfect. Okay, so thank you, Paul, again for this presentation. As you said, I'm a scientist here at the University of Southampton, and I'm trying to understand the origin and mechanism of lung diseases. And recently, last year, under the BRC, I've been appointed the scientific lead for the advanced cell culture model. The first action after being appointed in this program to create this network has been to create this group of bringing together people with the same interest in advanced cell culture system. And we have set up a network called SoCRATES, Southampton Cell Research and Tissue Engineering System. So it's a fancy name, quite nice. But before I start talking about what we want to achieve with the SoCRATES, I need to talk to you about Flash Stanley. Now, I don't know if you've heard about this story, but it's a boy that one morning wake up and has been flattened by a piece of furniture over his bed. He makes the most of it. He goes to adventures and he spends some time around doing a lot of nice things until he gets sad and upset about that. His brother restores his shape by bicycle pump. Why I'm telling you that? Because in the last 20 years as a research in the lab, I've done a lot of work using cells on plastic. I've been working as a Flash Stanley for a long time, where the cells are grown in plastic. It's not very physiologically relevant. And then the experiment has been done using this model. But now there is a more advanced research in which, like cells that derive from lung in my experiment, from plastic, they get lifted up and they are allowed to interact with each other. So not being attached to plastic anymore, they sort of differentiate in the way they're more physiological relevant for themselves. And they differentiate in what the cells are supposed to become. In the case of the lung, when the cells mature and differentiated, they produce cilia. So because they are free in their media, they actually float around. And they're actually quite nice to follow them around. I'm actually quite happy that I'm working to lung that allow me to have these very nice images. And the methodology I've developed allow the cells to have the apical on the outside, and that could be useful for downstream experiment that we also do, and I will explain a bit later. So we can use this methodology for drug testing, for understanding how the disease progresses and how can we stop them, and try to understand how the virus are able to infect cells and how can we stop to do that. Now, that little mouse in the corner is just to remind me that this methodology allows us to avoid using animal model for this sort of testing, because sometimes mice they're not really physiologically relevant. So we can save on using animal model for the experiment we want to do. There is a lot of work to be done. There is a long road ahead, but we've got a lot of interesting data. So at the top, what I'm showing you is image done by immunofluorescence, in which if you see on the top left, I've got, for instance, one of the spheroids we have developed, in which the blue in the center indicate the nuclei inside, the green stain for the protein that create the cilia, and in red, the stain for the protein of the mucus that the cells produce typically in the lung. And then there are other examples of other experiments we have made. But then another interesting slide that I'm going to show at the bottom, I've got also showing a system in which I've put together different cell types. And I've got like markers that indicate that these different cell type proliferate, differentiate, and create a sort of compartments that is typical of the lung tissue. And for instance, on the bottom right, I'm just showing a typical picture of a lung tissue directly taken a stain, and that's got similar characteristic of what you see in the organoids. Now, apart from nice images, this methodology allows us to use it to understand different disease. And for instance, in this slide, I'm showing you how the application of the different type of organoids we already have and we are using for understanding better. Like we can understand and investigate better lung fibrosis, COPD, chronic obstructive pulmonary disease that create a lot of changing in the lung and cause like severe chronic long-term diseases. We are using these spheroids to understanding tuberculosis, and also in some advanced model in collaboration with engineers, we are able to create setting up models in which we can understand asthma. This is just one of the few things that we want to do and we want to go forward. And that's it from
Mirella, very sorry.
Yes.
This is all, completely not anything I know anything about. do you mind going back to the slide before this one and just taking me through that again? Because I wanted to properly understand, well, not
Yes,
properly,
yes.
but
Yeah,
have a
of
bit
course.
better understanding of what I'm looking at there.
Okay, so at the top, I've got 3 images of organoids that we have developed using only the progenitor of the lung. So if you have tissue that derive from the lung, and disaggregate the tissue in a small little cube explant, like imagine you are actually, it's almost like disaggregating to a point that the tissue is just like almost like dissolved in a solution. If it's left in right condition on a plastic with the right media, the cells proliferate, and you are able to have a big amount of cells. But as I showed earlier, they are just attached on plastic. And the condition that I use, the cells are there dividing without not any specific characteristic of the lung of origin, because they are just progenitor. They can become cells with different functions that are in the lung, but in the way in which I grow to start with, they are undifferentiated. They are, as I show, sorry if I have to do on the left-hand side, can you see the image of like cells that have been just disaggregated, left in the media for a few days, and that is what they look like. Then you actually are able to, we can use protein enzymes that detach the cells from the plastic. And once you've got them in a solution, we see them in another media with a lot of supplements, where the cells are free to differentiate in what they will become, like in differentiated mature cells. And if you do that in a way that the cells cannot touch the plastic, it cannot remain attached, the only option they've got is to interact with each other. Once they interact with each other, they sort of stimulate, they connect it all to each other. And depending from the viscosity of the environment, the condition, the supplements that you give, the undifferentiated progenitor cells mature and differentiate in cells producing mucus, typical of what we've got, like when you got cough, you produce a lot of mucus because there are goblet cells called, they produce this protein. And also you get cells differentiated with the ciliated, so they produce cilia that helps the mucus that trapped the pathogens, and any dirt that can come in the lung can secreted and removed by a cough. So I'm able to create these small spheroids in which I can have representation of cells typical of the lung in the some of the cells, because anyway, there are 40 types of cells, but these are the most representative that usually are used in the model. They are actually the first line of defense when there is an infection in the lung. And a lot of virus use these cells as a way of entry. So if you got a model with this type of cells, you can actually have a model to study the infection of flu or COVID or rhinovirus entering the lung. And they float and they swim around because the entire surface has got the cilia on the edge, and they are able to, okay, it's not floating anymore. Anyway, and they swim around and they taste the environment. Anyway, those organoids, they can be taken out and they be fixed in a way that you can preserve the structure, and they are embedded in a paraffin in a block, so you can cut very small sections, and they are put fixed on a glass slide. In this way, you can, if you want to identify for a particular type of protein, characteristic, for instance, the protein that defines the cilia in the organoids, you use antibody connected to a fluorescent molecule that is going to find the cilia protein will be found by this antibody and will stick to it. And because every antibody has got also a fluorescent molecule on it, every time there is this structure of the cilia protein plus the antibody plus the fluorescent, I will be able under the microscope to see every cell that has got the cilia because it's attached with the antibody with the fluorescent molecule on it. And I can choose different colors. So I've chosen the color green attached to the antibody that is recognizing the cilia on a section of my organoid that has been previously cut and prepared on it. Then the red is associated with the antibody that will recognize the mucin, and the blue is just a dye that stain every nuclei in the spheroid. In this way, I'm able to have a characterization of the protein I'm interested in. The one in the middle is similar, it's just another organoid. And the one on the right-hand side, P16 and P21, are two proteins that are associated with senescence, so the aging of the cells. And I wanted to see if I can identify also senescence in the cell. And
those are combined. The colors are combined in that one, whereas they're very distinct in the other.
Yes, it's because P16 and P21 are both markers of senescence, and they are both present in the same cells. So when you have got two different proteins in the same cells, the antibody will be next to each other, and because you combine the red with the green. I'm sorry, P21 is actually, it should be green, but it's a big golden. But red and green under the microscope, when they are on the top of each other, they look like sort of yellow. It's like the color is different because there is this overlapping. And because the degree of the presence of one protein compared to another is not the same in every cell, because there is a different level of senescence, you see almost like a range of the different colors between green to red to yellow. You can even increase the number of markers. You can change the color. You can have more than two, and you can identify the different cell type, the different protein that you are interested. And because for a lot of cell type, we've got also a marker specific to recognize. You can do a sort of cataloging, so you can check which type of cells you have got in your spheroid using the different markers that you know. And you can sort of create almost like an ID and say, okay, these spheroids, these organoids present the following markers. So I can quite confident that this type of cells are present in my model. At the bottom, I've got there are organoids in which I've put together not just the progenitor cells of the lung, but also fibroblasts. Fibroblasts is almost like support cells that you find in a lot of tissue. They've got a lot of function, a lot of roles, and they are different cell type. What is interesting in my methodology is that the fibroblasts that usually act as a support in the tissue, in this structure, create the core on which the progenitor cells stand on, and while the progenitor cells differentiate in cells of the epithelium, the fibroblasts stay in the core as a support. And I can recognize them because I've used a marker called vimentin that stain fibroblasts, mesenchymal supported cells, while the cells on the edges, indicated just by the nuclei stain blue, don't have any mark, any color. While if you look on the right-hand side or the center, you see I've used keratin 5, KRT5, and that is a marker of the epithelial cells. It's actually staining the cells around, while the core is empty, hasn't got any staining. And tubulin, this time is labeled with the red, is staining the edge where the cilia are. And
sorry, and so with these things, are these I don't know the word, the processes that would take place similarly in vivo to in vitro?
Okay. In vivo, the differentiation the differentiation in, sorry, I'm not sure I've understood, but I'll make a comment. Please tell me if
Sorry, I didn't explain my question well enough. What I'm just thinking is you're doing this in a lab, and you're creating this and you're seeing these effects, and I'm just wondering, are these things that you're able to create very similar to what is actually going to be going on in a body in this situation, or is it different in the way that your 2D would be different?
Okay, in vivo, the orientation and the relationship between the two different cell types is very similar. But we are talking about in a tissue, in a lung in particular, there are 40 different cell types. And we are talking about the trachea, the bronchi tree, and in every lobe that is connected with the alveoli, and in the peripheral part of the lung, and I've got other cell type. I cannot recreate a lung, a mini lung, because we don't have the technology with this method to recreate the bronchial tree and the alveoli. This is a methodology that allows me to recreate a bit of the environment of the bronchi with the structure underneath, and allow me to recreate the environment of the epithelium that is between the environment when we breathe in and the tissue underneath. I don't have vascularization, so I don't have the cells. So I cannot recreate the entire system like that. There are different, yeah.
Mirella, shall I just come in as well?
Please, yes, of course.
I think that's a really good question, and it's exactly the question which we often get from our scientific reviewers when we go externally for funding. And I suppose the question is what, we can't recreate the whole lung, but what is the minimum amount that we can recreate, so then we can replicate the processes going on in infection or in disease, so we can then study them in the laboratory. And for example, what we've done in tuberculosis is we have made these little spheres, infected them with tuberculosis, and looked at all the changes in genes and compared those changes in those genes to what happens in patients. So when you take tissue from a TB patient and you look at the uninfected versus infected. And we've shown that if you look at what's going in the old traditional flat Stanley 2D, there's very little similarity. Once you make it into 3 dimensions, it's more similar. And when you put it into 3 dimensions with collagen inside, so the main sort of matrix fibril which holds the lung together, it's even better. But it's still only about 80%, but you progressively get closer and closer to what's going on in patients.
So it's an approximate, there are steps of approximations, yeah. I guess, and yeah, thank you.
And I think the final thing I'll say is part of Mirella's work for the next year is to repeat that process for influenza infection. And so say, well, can we also do those steps forward? And for each disease, you have some information about what's going on in patients, and we can sort of use that as what we call the ground truth. And we move the models forward slowly towards the ground truth. And as you get closer and closer, the more useful it then gets for developing drugs and things.
Yeah, and Paul, if I could add as well, you could actually recreate a model that are more specific for the part of origin of the lung. So you can recreate model that derived from cells from the nose that got a certain characteristic. Then you can also recreate organoids that are coming from the alveoli, from the extreme external part of the lung, and they've got different structures because there are different cell type that make it. And you can ask different questions because, for instance, the organoids that come from the alveoli side, they can be used to measure pollution, to understand the role of the pollution in the cells. And the organoids that I'm trying to set up, there could be a good opportunity to understand how the infections work, viral infection or bacterial infection work, because that is the environment where the pathogen enter in the lung and interact with the cells, causing disease in most cases. Okay, and I think my presentation was just my next slide was just thank you. Any questions? So we were within that.
Sorry, I came in a bit early on that.
Please don't. I don't know. Any more questions?
Well, yes, thank you. I get Barney, do you want to turn or shall I carry on? No, okay. So yes, very interesting and something I really know nothing about. So I'm wondering several things. One is the more you can be looking at examining something that is as close to the thing that you're trying to find out about, the better. So you will have more applicable information. But we start.
Mice are the sort of really standard ones. Then you sort of get to rabbits, and then you get to non-human primates, and so you know, monkeys, because they're so close genetically to humans. There are both, you know, for me, I would say there are both ethical and expense issues of working with monkeys. But often the very last step before you do a human trial is to test in a small number of monkeys. But what you want to do, I would say, is do as much as possible in the simple systems before moving to that. And then in terms of the cellular systems, it's really the graduations which Mirella talked about, from old style 2D to increasing levels of complexity. And as Mirella alluded to, one can, you know, you can go more and more to town on this. You can say, I want to add this type of cell and that type of cell. And to give one example, there are engineers who make little tiny channels through plastic. They line those channels with blood cells, and then on the outside you put the other cells, and so you can flow blood-like material through and have your other cells on the outside. So you can go more, and you can layer complexity on complexity. And what we're trying to find, I suppose, is the sweet spot between sufficient complexity that it reflects what's going on in the patient, so that we can test against what's going on in the patient, versus it's not so complex that you can't really do, because often we might want to do 30 different conditions, and you know, we might test 30 different drugs, and so you need to be able to make a lot of these units, as it were, to test them. You know, just making one is no good, because you want to have your uninfected and your infected, for example, for flu. And then you want to have infected with drug A, B, C, D, E, and measure what's going on. So for me, there's a graduation of animal model systems. There's a graduation of cellular systems, which is where we're operating. And then once you get into patients, you have a graduation from just a very small study, see if the drug is safe, to a phase 1 study where you might give it to a handful of patients, to then doing 30 or 40 patients, then doing 400, 500 patients, and then into society. And I think, you know, the goal here is obviously around accelerating the throughput and increasing the hit rate.
Paul, if I can only add, sorry, if I can only add one more thing to what you said. Sometimes it's not a matter also to complicate our life and making the most complicated model just because we can do it. But it's also a matter of understanding the question that we want to answer and what is the best simple method that can help us understanding the problem and solve. For instance, in the case that I've shown you that our model has cilia. Now, if you've got to studying a disease in which the cilia are affected, like primary ciliary dyskinesia, that can create a lot of problem in patients with that disease. And you have got a compound that it may improve the quality of life, improving the cilia structure. A simple model with cilia will suffice, because I don't need to have an entire lung to study that sort of question. Or if I am investigating a viral and entry, the point of entry for a virus, a respiratory virus, and I know has got entered to the trachea, I will have a model that has got component of the trachea. And I don't need the endothelial or the rest of the lung to answer. So it's also a balance, try to improve our methodology to answer better question, but also keep it simple. To the simplest way to have a simple readout, because the answer has to be simple. And then you can build up maybe in a more complicated one, and you can use maybe in some circumstances animal model or more complex system, or increase the numbers.
Thank you. And so if what we're looking, if what we're, you know, if the objective with something is, okay, we're looking to find a treatment for this disease, people have, you get the drug development, and then it is tried on people. There wasn't always tissue engineering, so people developed drugs and they tried them on people, presumably knowing something about what they were doing, but with not this sort of information. Do you see, or to what extent, I mean, because I know nothing about it, to what extent is tissue engineering either now or going to be something that you kind of have to have in place in order to be able to do, to get through the hurdles to get to a trial for something?
And I think that's a very good question. I suppose the first thing to say is that probably about 1 in 10 drugs succeed when you get to the clinical trial stage. And so given that it often costs £1 billion to get to that stage, there's a huge amount of waste in the drug development pipeline. So one thing which we would hypothesize is that going through a tissue engineering stage would increase that hit rate, and so would de-risk before that expense. It's very notable that last year the United States Food and Drug Administration changed its rules, saying, so since I think about 1946, every drug has to go through a mouse model. And it changed the wording, saying it must go through an in vivo or advanced tissue system. And so the United States Food and Drug Administration has said that you can proceed to a clinical trial by using the kind of system which we're talking about here. I think a lot of people have some concern about that, because I think you would still want to give it to a mouse to check for sort of safety and things. But certainly what we're proposing in Southampton is in line with global trends, and you know, for the Food and Drug Administration to make that change is important. I suppose the proof of the pudding will be in the eating, about whether, if drugs have a higher success rate because they've used this stepping stone, then we will know it's true. And when one considers the difference in the immune system, for example, between mice and humans, I mean, it's so massive. It's not surprising that having a human system should improve our hit rate. And so we're not saying we're going to replace animal systems completely, but I think we can replace them quite significantly. And then the compounds which come out at the end have a much higher chance of success.
Thank you. So yeah, then I'm thinking about cost. So you need to harvest these cells from people. Well, just a quick question, and I wish I didn't get to keep switching my microphone off, but where are they? How do you do that?
I can talk, Paul. There are, okay, we have got one good thing about this research in Southampton, there is a good, strong collaboration between scientists like me and also clinicians involved in surgeries. So when people come to surgery for a procedure involving the lung, for resection, for different type of reasons, there is always, when there is samples or tissue that can taken out, it goes to pathology because it gets processed and analyzed. So to answer back to the surgeons for the disease status, for information regarding the patient. But a lot of tissue can actually go to waste, or can what we got with under the ethics that are already set up in Southampton, if the patient consent, we can actually get access to that tissue that otherwise will be destroyed. And that is where I get it. And there is no cost, like huge cost involved into it, because it's just the cost related to the infrastructure that allow the tissue to come from the theater to us. And then it's the cost associated in the lab, but it's not an additional huge cost that has to be added on the top for doing research. It's actually, I have to say, it's quite economical way. And it could be good because with this connection between the surgeons and the surgeon on board in our research, we get access to lung in a way that this type of analysis research can go forward quite frequently.
And so if people, people need to give permission for consent for that to be taken. Do you know what percentage of people give consent?
I don't know the data, but I know the way in which it's set up is there are the nurses that help us give access to the tissue. They every week on Friday usually, I get an email saying next week there are in the list 3 patients, and they give me the date. In the week after, and then when the samples, I will get the sample on the day when the surgery has been applied. And I have to say, I got the list on Friday, and the week after, if there are 3 samples, 3 people having surgery, the week after, usually I get samples from at least 2. And sometimes I don't get it because it's too late or has been postponed. So I would say it's a quite high level. It's quite rare that I've heard that people haven't consent, because I'm asking for tissue that is going to be destroyed, and that the ethics is quite clear in what we wanted to do. And also there is the samples also get used for understanding cancers, because the tissue that gets taken out also, there is cancer tissue that goes forward also for research.
That's good, because it sounds like overall in this situation people aren't too concerned about the ethics surrounding that, the use of their tissue. So that's helpful, isn't it, I guess. In when you get the cells, I'm wondering as well, is there how much of a lifespan do those have? Do you stick them in a deep freeze and they last for as long as you want, or?
Okay, the cells in culture have got a limited life. So just briefly about the methodology, the cells go into culture on a plastic, and I will have them growing up to a couple of weeks. After that, they grow the space and I need to take them out. So I use an enzyme to detach them from plastic, and I can put them again in another bigger plastic or put less on them. I can do this step up to 6 times maximum. After that, the cells run out of steam, like they become senescent, they don't replicate anymore. What I can do, what we call passage, like every time I detach from plastic and move away, I can take a little amount and put in the freezer, -80. Liquid nitrogen, actually even lower, and I can keep them for years. But I can take them out start again from whatever passage I froze them, I will have a passage +1. But I cannot do that forever. I manipulate them genetically to make them immortalized. And there are ways in which I can avoid the telomere or the chromosome getting too short, so they can be kept in culture for longer. And I can do some manipulation on that level, and they can last longer. But the type of work I'm doing at the moment is based on finite number of lifetime of the cells I'm using. But yes, they can get immortalized.
And I think that's a really, I think for me, that is the key question of this discussion actually is, and Mirella, maybe we should make a little note to try and prospectively collect how often people consent and don't consent, because I think that's a really good thing. We need to know that information, don't we? Because if 80% of people are declining, then there's something worrying. If 80% of people are agreeing, then most people are happy. And most, you know, to date, most people are happy to say, essentially the view is, well, you're doing an operation on me. There's some tissue which essentially is going to just be incinerated. I'm quite happy for you to grow some of it for a fixed period of time in the lab and do an experiment, which probably won't help my own care, but may help people in the future. And there's that sort of altruism. And I think that one of the huge advantages we have in the UK is the NHS. And because the NHS is free, people are happy to donate their tissue. Whereas I think if you're paying for it in the States, you might have a slightly different interaction with your surgeon. You might go, well, I've just paid you a lot of money for this operation, and now you're asking me to keep the tissue. I do feel it's more difficult if you then take the cells and you genetically modify them so they live forever, because that's a slightly different thing you're agreeing to. And I'd love to hear your thoughts about that and about how we might explore it.
I do think, I'm just sort of thinking, if I said to you now in a sort of Frankensteinian way, what do you think, because I don't know anything, what do you think the potential of this is? Now you told me that the people are giving their consent, and they're giving it for a very specific purpose that you have told them. So I think if you've got a good take up, it must show that this how specific you are with what you're saying you're going to do, and the fact that that's not something worrying is credit to you, because that's an important thing for people to understand. How do you guard against something that is kept for a long time, definitely only being used for precisely the purpose for which you said it would be used, if technology progresses over that period of time? I think that's kind of difficult, and I don't know what the Frankensteinian future could potentially hold. What is the limit of that?
Bonnie, do you want to come in? I see you've got your hand, but because I mean, I think this is, you know, I think that's really useful feedback, and this is the nub of it. I mean, I think the short term, what we're doing now, I think people are happy with. You know, the Frankensteinian thing is you genetically modify the cells, and then you create a thing called an induced pluripotent stem cell, which is almost like an embryo, and you could make a new person from it. Now, we're not proposing to do that at all, but even just saying to someone, well, we're going to modify your cells, and we may be doing an experiment on them in 10 years' time, looking at a new treatment for a new pandemic infection or something, it's just a different conversation, isn't it? We're going to do these experiments, and they're all going to be done and dusted within 6 months, whatever. And Mirella, maybe what we need to take away as a second action point is to really be clear in our mind the difference between the sort of the hot experiments where we take the cells, we process them, and ones where we archive cells, and to think about how we make sure we monitor that archive, and we probably need to have an external inspectorate of the archive, as in it can't just be the people doing the experiments. It perhaps needs to be I'd be interested to hear whether you feel that partially addresses it. So if every 6 months the archive is checked by someone who is not directly involved, they can keep an eye on making sure that we are disposing of tissue appropriately, and we're not holding on, for example. So perhaps we need to get consent for long-term freezing versus hot experiments, because there's a difference.
Yeah, I think.
Sorry, carry on, Barney.
I was just going to say, I think that's probably an important distinction for me, because I mean, in this area, I'm as lay as anybody else. I was trying to understand your explanation as much as Sian, and trying to boil down what you've been saying. I've heard a lot of positive things about, possibly moving away from, I wouldn't say reliance, but sort of moving the spectrum away from vivisection to working with human cells more, and the idea of actually taking the cells, well, it's no more invasive than the operation that presumably I'm having. But the idea of giving cells and not knowing to what purpose they might go to, it's strange. I haven't been through that process, but I would imagine that even though I'm giving you the cells, I'd feel some sort of ownership over them. They're still my cells, and I may be comfortable with Paul and Mirella using them, but if they're going to be archived, who's going to use them subsequently? What purpose are they going to put them to? And the other comment that I'd make is when I was involved in something around the vaccinations and hesitations around vaccinations, and it was with a predominantly Muslim audience, and they had heard that the vaccine had been tested on pig cells, cells from a pig, and actually making the explanation of where the cell has come from, what it's been tested on. No, it's not haram. Yes, it is fit for humans. That was actually quite a difficult explanation. And I think making that as simple to explain as possible is a good thing. So yeah, I think that distinction between what you're using it for now and what you might be using it for in the future, and actually being able to quantify what it's been through up to that point, is quite important. Does that make sense? Yeah.
Yeah, because I think it may be that from an ethical standpoint, even though physically it's possible to keep things in liquid nitrogen as long as you like, there may be an ethical discussion to have around how long these things should be kept. And people are, I think your point, Paul, about things that are done in the States being a bit different in terms of how one might look at the motivations from here. What's going to happen to the NHS? So would I feel differently about whether I'm happy for people to be using my things if there's not an NHS, and if I know that everything is sort of profit driven in my healthcare? I might feel a bit differently about it then. So yes, I think it's one thing to give consent for the present, and it's quite difficult to give that for the future, when, as with an increasing number of things these days, seemingly it's something that could be misused by somebody, possibly, or not used in a way that I would want to support. If I am absolutely opposed to the development of people from my stem cells, well, I don't want to be a part of that. So I don't want to have supported it by supplying my tissue. So yes, I think those are discussions that almost, if you're saying we're going to use it, you almost need to be thinking, and we wouldn't be using it in these circumstances, this would not happen. And then yes, having somebody external who's going through and is checking the validity, the usefulness of the protocol that you've got. You know, that's the thing that needs to be. The protocol is as it is. I've given my tissue now. That's the protocol you've got to stick to with me. But on a regular basis, you may be renewing your protocol. And for the new people who come through, they may have slightly different conditions under which their tissue is needing to be kept. And then just sort of in terms of thinking about diversity with people giving their permission to use this stuff. I didn't know with your information, is it something that is mostly done through spoken word, that, you know, you say, oh, well, we're wondering if we could have permission to blah, 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 and here's the paperwork, but actually you've talked them through it? Is that how that usually goes? And so language barriers and comprehension barriers hopefully are things that are addressed and overcome successfully because you've actually got a person, not just a piece of print. Is that right? Yeah, so that's good, because I always think that's a concern, but that's obviously, that's really good, that's taken care of.
Yes, Sean, if I can say, yes, the consent, there is a person, could be the surgeon or could be the nurse that go there with a consent form. And yes, it's a piece of paper that explains, but it gets communicated verbally, like there is a contact between two people. And also it's not done in a way 5 minutes before the patient is wheeled in the surgery room. It's done the day before to give time. I think there is an interval of several hours. I think it's at least a day between asking for consent and giving and the surgery. So you've got plenty of time. The consent could be withdrawn even after. And also, if the cells are collected with the purpose that is stated in consent, the things cannot change. Although maybe there could be slightly changing the protocol in which I handle the cells, but the aim of what I'm going to do is always the same. I may improve the protocol, but the system is always there. Even if I leave the lab in 10 years' time and there are cells in the culture in liquid nitrogen, there won't be another person they say, oh, they are there, I can use it and do all sorts of experiments they were not designed to in the first place. So this is not going to happen. Everything will be open on what will be done based on what was approved in the first instance.
Thank you.
But Mirella, maybe we should note one more action point, which is to consider a structured consent form, which I'd be interested to get your feedback on, which says, I'm happy for my cells to be used immediately, A, B, frozen for future use, C, genetically modified so that they're immortal, or D, shared with the pharmaceutical, in collaboration with the pharmaceutical industry, because there are different touch points, aren't there? And so in a way, A, most people are happy with, but one could see there might be a sort of cadence of uncertainty, and the sharing of the pharmaceutical industry comes out of Sean's point about if the NHS becomes commercial, then actually these cells start having a value, quite a significant value, and I don't think people would be wanting to give their cells so someone else can make a profit from it.